Now, continuing on with our discussion of protein synthesis, we come to the stage 3, which is the peptide bond formation, of the peptide bond, also sometimes just simply referred to as elongation. And I want to go through this. It actually also has 3 steps, and we'll talk about the 3 steps of stage 3 as well. And so here you have a set of requirements. For one, you need this initiation complex that we formed in the stage 2. You'll also need a set of charged transfer RNAs. So you're going to need basically a full set of these guys. And you'll need for all 20 amino acids, you have to have the charged transfer RNAs. Now here we're also going to have some elongation factors, and we're going to just shorten them and call them TU, TS, and G. Yeah, but these are, well, for EFTU and EFTS, but these are elongation factors, which basically we just abbreviate as EF for elongation factor. And you will also need GTP, because GTP is going to be a part of the addition of every amino acid is going to require 2 GTPs to be hydrolyzed. And so peptide synthesis takes quite a bit of energy. Every amino acid that gets added, you hydrolyze 2 GTPs, and we'll show where those come in. So to get to the first step, so in this first step is the binding of the incoming amino acyl transfer RNA to the A site. So basically, here we are in step 1. We're going to bind this amino acylated transfer RNA. It gets bound with the assistance of TU. So this entire complex is what gets recognized by the ribosome and is bound by the ribosome. So the ribosome binds this shape. And when it binds that shape, it's a little more complicated than just that. So what I want to do is, there is another elongation factor that is involved in this step, and we don't really see it unless we go to this next slide, and we see what is referred to sometimes as the TU-TS cycle. So the TU-TS cycle, basically what we get is when basically when GTP is hydrolyzed, you get TU-GTP bound. And so to get GTP bound again to TU, you have this elongation factor TS, and it will exchange for the GDP. So TS will bind in place of GDP, and then GTP will bind in place of TS. And so the TU-TS cycle is really just a series of steps that allow for exchange of GDP with GTP. And it's interesting that you need an entire elongation factor to accomplish that. I'm not sure why it is that, but this second elongation factor is just simply to allow for the exchange of GDP with GTP. Now once that takes place, the EFTU is charged again. This is the activated guy. It can then bind to our incoming amino acyl transfer RNA, giving us that we need for step 1. And then one last thing I wanted to say about this slide is just that when you have the GTP hydrolysis, so GTP hydrolysis gives rise to a conformational change that's referred to as accommodation. So this accommodation is basically what's being illustrated right here. You have this amino acid part, this amino acid attached tRNA arm is going to swing over into the active site here. And so you get the combination is just a conformational change of this transfer RNA to put the amino acid into the active site. And that positions the amino acid into what is known as the peptidyl transferase active site. And so now we come to step 2 in this process, which is basically where we have peptide bond formation. And the peptide bond formation is not really that complicated, but it's interesting that the details of it are fairly well understood. So here what you have is you've got the amino end of the incoming amino acid performs a nucleophilic attack on the carboxyl of the ester linked in this case, it's just a single amino acid, but in the case of a growing polypeptide chain, this could be a polypeptide chain out here. So, but here, we're just forming the first peptide bond, so this is just our F-met. And so what happens then is this, everything that's over here in the P site gets transferred over to the A site. And so that's what you wind up with here. Now, this is our transition state. It's an oxyanion, and this will be an unstable transition state species. And so what will happen is basically the energy of this has to be lowered. And actually this takes place, this portion of the ribosome is actually the 23S ribosomal RNA surrounds the active site. So the catalytic activity, the lowering of the activation energy, or the lowering of the energy of this transition state, is basically this guy gets stabilized by interaction with the ribosomal RNA. And so we get promotion of this process by lowering the activation energy, and that all takes place in the ribosomal RNA component, not the protein component of the ribosome. And the ultimate end product then will be our new amino acid gets added on to the carboxyl end of the peptide chain. But you'll notice how the transfer RNA, then the A site, has shifted in conformation over to the P site. And that's because the growing polypeptide chain has a channel that it will pass through to exit the ribosome. So this channel, the growing polypeptide chain, will basically just get pushed out this channel, and so the A site transfer RNA moves over into the top of the P site, so that the polypeptide chain is continuing to go up the channel. But then we still have a problem. We need to move the entire transfer RNA over, and so that leads us to our second step, which in the second step we've got what is known as translocation. So here, translocation actually has a couple of sub-steps, but it requires this elongation factor G. Elongation factor G will bind to the ribosome at this location here. So it binds to the A site transfer RNA. When that occurs, it basically will, and it also requires that it gets bound with GTP. So the binding of the elongation factor with GTP will bind to the A site transfer RNA. When that takes place, we then will have a second sub-step, which is GTP hydrolysis. When GTP hydrolysis takes place, that causes it basically, like, you can think of it in two ways. You can think of it as the ribosome gets pulled over. That would be a pulling view. And you can also think of it in this way, is that the messenger RNA gets pushed over. So basically, in other words, the ribosome moves towards the EFG, so that the EFG then is located in the active site, or the A site. And corresponding to that is a conformational change of this transfer RNA as well. But basically now we have the entire ribosome has moved over by three base codons. So right now we have the second codon has moved over to the P site. And to complete the translocation, we actually have the dislocation of EFG, and in part you can kind of imagine that once this conformational change has taken place, the binding affinity of the EFG is no longer very high, and so it just comes off and it leaves an empty A site. And then of course after this, after the A site is vacated, the next amino acyl transfer RNA will be loaded via the EFTU mechanism. And there's one other topic I want to hit on with regard to the EFG, And that is structurally, because structures have been determined for these guys, the EFTU structure is actually very similar to the EFG structure. So EFTU, EFG has a very similar sort of shape, and it basically mimics the anticodon loop here is mimicked. The anticodon loop here is mimicked by this in shape and in charge, and so that helps its binding to the A site. And so the overall shape of this guy binding to the A site is facilitated. And this is, now remember, this is transfer RNA. This is transfer RNA here, but this is protein. So that's what's kind of cool about this is actually a protein that's shaped like the tRNA, and helps with this translocation process. Now, one last topic I want to hit on with regard to elongation, and that is that we see a lot of, as we mentioned before when we talked about the synthetase, we see a lot of proofreading. We even see proofreading in the synthetase. Well, it turns out there's also some ribosomal proofreading. And so it turns out that the GTPase activity of the EFTU plays an important role in the placement of the correct amino acid in the growing chain. So remember, EFTU has to hydrolyze GTP, or GTP hydrolysis has to take place in order for the incoming amino acyl transfer RNA to get loaded correctly into the A site, and even in the correct conformation, the accommodation conformation. Well, it turns out that the EFTU GTP complex and the EFTU GDP complex, so basically before GTP hydrolysis and after GTP hydrolysis, both of these complexes are bound to the ribosome for just a few milliseconds before you get the final dissociation. Now, this short period of milliseconds allows for codon, anticodon to be proofread. So what happens is, if the incorrect tRNA is bound, it can dissociate during this time. And so if the correct anticodon-codon pairing does not take place, then the tRNA will just dissociate, and you'll have the correct one, EFTU tRNA, put back in place. Now, what's interesting is that the way this has been demonstrated is the use of this GTP analog called guanosine 5'O'3-thiotriophosphate. So basically, this looks like GTP, except that our gamma phosphate here has a different structure, has this thio group in place of an oxygen. And so what this does is this leads to a reduced hydrolysis rate. And the interesting thing about that then is, if researchers who use this to study this process, they found that if you use this instead of your normal GTP, you get a reduced hydrolysis rate, and you get higher fidelity of correct amino acid incorporation into the protein. The problem with this higher fidelity, though, is that it leads to a slower synthesis rate. And so what you have with GTP binding, this millisecond time frame gives you enough of a brief period of time to give you proofreading opportunity without slowing down the actual the overall process, the overall rate of peptide synthesis or protein synthesis. So this is kind of an interesting little aside, and the use of this guy to demonstrate that was important. Now this brings us to the fourth stage, also known as termination. And you remember there are 3 stop codons, and these are UAA, UAG, UGA in RNA, and then in DNA we have TAA, TAG, and TGA. These are the, typically we're going to see these guys when we're dealing with DNA, so that's why I wanted to make that connection. But in RNA, they're these guys. And so there are these 3 stop codons, and in bacteria there are 2 release factors. And there's also a thing known as the ribosomal recycling factor. And so the ribosomal recycling factor is this guy right here, ribosomal recycling factor. And it turns out that termination then takes place in 2 steps. We get our release factors, the RFs. The identity of these guys actually depends on the stop codon. There's 2 of them, and the 2 recognize different stop codons, and I'm not going to go into details on that, but basically there are 2 of them, and they will recognize different stop codons. But the release factor binds to the stop codon and in the A site, and leads to the hydrolysis of the ester-linked polypeptide chain in the P site. So we're going to get hydrolysis of this guy. And when that occurs, then basically we've got polypeptide release. Now, what will then subsequently take place is now we've terminated peptide synthesis, but we want to recycle. And so to recycle, there actually are a couple things that will take place. We've got initiation factor 3, and we've got this ribosomal release factor here. That initiation factor 3 comes on, and then basically we get loss of our EFG, and then we've got release factor with, what am I saying? I back up there, sorry. I meant to say we have EFG with GTP bound together with initiation factor basically associate with the ribosome. Then we have the ribosomal release factor associates with the ribosome, and the hydrolysis of GTP and the association of the IF3 are basically all done kind of together. But what was assumed then is that EFG then causes this translocation of the release factor, and once the release factor is pushed into the P site, then basically everything dissociates. And as everything, when you get this GTP hydrolysis taking place here, that pushes everything over, pushes the ribosome over. Everything then dissociates, and the initiation factor 3 binds, preventing the reassociation of the 50S. And so then the ribosome can reassemble at a start codon, ribosomal binding site, and carry out another polypeptide synthesis. And so you actually wind up with this complex at the end, and you get the release of all the other components, the messenger RNA, and so forth. Now, one topic kind of related to the overall process of actual peptide synthesis, protein synthesis, is what we've talked about in these first 4 stages, is starting and then synthesizing a polypeptide chain. And basically what you get actually, and this will occur both in eukaryotes and in prokaryotes, although in eukaryotes you don't get this complex that you see down here or you don't get the DNA as part of it. You can have a single message that has multiple ribosomes associated with it, like this. But you won't have multiples of these. Well, these are sometimes referred to as polysomes. So this would be an individual polysome. This basically has multiple ribosomal units synthesizing polypeptide chains, all at various stages of polypeptide. So this guy here has just initiating. You have a short polypeptide chain, you have a long polypeptide chain here, and so this guy is one entire polysome. And you do see polysomes in eukaryotes, but you don't see like this in bacteria, where you actually have polysomes kind of linked together. and these linked together polysomes. This is actually an electron micrograph up at the top, but these polysomes are linked together because the RNA polymerase basically is here in these sites and is allowing for multiple polysomes. You have like an RNA polymerase associated here, an RNA polymerase associated here, and so forth, with each of these polysomes. So each polysome has an RNA polymerase associated with it, because you're going to, for each polysome is basically one big long line of message RNA with a bunch of ribosomes associated with it. And it's just kind of an interesting observation, is that this allows for a much more efficient synthesis of protein. So if you've got a message, you've got a transcript, you can basically, from that transcript, make bunches of proteins at the same time. You don't have to wait for one to get finished before another ribosome binds to it in prokaryotes. And apparently this also is able to take place in eukaryotes as well, although I'm not sure the mechanisms for that is quite as well worked out. Now next time what we'll do is we'll go on in the next lecture. We'll begin with stage 5 in class on Friday. We'll talk about stage 5 and the post-translational processing. Post-translational processing, we will talk about that and how proteins are processed after they've been synthesized as kind of the stage 5 of protein synthesis.